Okay, the final section of the textbook is on nucleic acids. And there are two main categories of nucleic acids. Those are DNA and RNA. And they both have extremely similar structures. So DNA stands for deoxyribonucleic acid, and RNA is just ribonucleic acid. Or ribose nucleic acid and deoxyribose nucleic acid. I forget if there's an S in there or not. Anyway, ribose is a pentose, so it is a 5-carbon sugar, and the deoxy in DNA means that the OH group on the second carbon there has been removed and just replaced with a plain old hydrogen. And the ribose, or the deoxyribose, is going to cyclize, and then it's going to have this phosphate unit attached to its 5-carbon, and a nucleobase attached to its 1-carbon. So it is a sugar, but it's a modified sugar that's going to perform very specialized functions in the body. So that is what a nucleotide looks like. A nucleotide is 1 phosphate, 1 ribose, and 1 nucleobase. So what is a nucleobase? There are 5 different kinds of nucleobases, and they're usually represented with a single capital letter. So adenine, thymine, guanine, cytosine, and uracil, represented by A, T, G, C, and U. So DNA contains adenine, guanine, cytosine, and thymine. RNA replaces the thymine with uracil. We'll get to see what they look like here. They're conjugated ring systems with nitrogens and often oxygens as well. And that's going to give rise to a lot of lone pairs and a lot of hydrogens bonded to highly electronegative atoms. So there are a lot of opportunities for hydrogen bonding. You can see that the thymine and the uracil are extremely similar to each other. Before we move on, actually, I'm going to go a little bit outside of the scope of the textbook here and talk about RNA versus DNA. So looking back at like the microscopic organisms that seem to be the oldest in the evolutionary tree, it's become pretty clear that RNA predated DNA, and that at some point there was a mutation or a change or something that gave rise to DNA, but RNA came first. And so they're very similar to each other. They are both very, very old, like early days of life type molecules. You're going to see that RNA in particular is really versatile. It's used kind of for everything that else. It's used to bootstrap all of our other cellular processes. So the RNA is used to make proteins and other things in a way that is kind of interesting. And that's because RNA was one of the first biomolecules that were incorporated in complex life, and so it had to serve all of those functions. Anyway, okay, so here we see the chemical structures of the 5 nucleobases. You don't need to memorize these at all, but I do want to point out a couple of things. So I'm going to turn my mouse into a pen. Okay, so here on the adenine, we have a hydrogen bond donor and a hydrogen bond, oops, nope, that's not what I wanted, and a hydrogen bond acceptor. Well, it wants to make that a line, but that was supposed to be 2 dots. Then here on guanine, we've got, now what's it doing? We have a hydrogen bond donor, a hydrogen acceptor and 2 donors. Cytosine, we have a donor and 2 acceptors. And then thymine and uracil both have a hydrogen bond donor and acceptor. And when I say that, I mean that the hydrogen bond acceptor is going to have a lone pair on an oxygen or nitrogen. The hydrogen bond donor is going to have a hydrogen bonded to a nitrogen or an oxygen. So you see the guanine and the cytosine, they each have 3 sites, and they're even like kind of inverted. The guanine is sort of a convex trio of hydrogen bonding sites. Cytosine is kind of a concave trio of hydrogen bonding sites, and they're going to fit together neatly, as we will see in a minute. So not only do the nucleotides have the phosphate and the ribose and the nucleobase, pardon me, but nucleotides can bond together. That phosphate group can bond to a neighboring nucleotide and make what's essentially a polymer of these nucleotides. And so you'll have these chains with different nucleotides, one right after the other. And that's what makes the nucleic acids as we know them. And so nucleic acid chains will come in pairs, and they will bind together by these hydrogen bonding interactions. So adenine and thymine are going to have those hydrogen bonding interactions right there. And then cytosine and guanine are going to have 3 hydrogen bonding interactions. And you can kind of have these in whatever sort of order you like for one of the strands, but the other strand must complement it. And so if we look at the strand on the left, starting from the 5' end and going to the 3' end, we have adenine, cytosine, guanine, and thymine. Well, the complementary strand of DNA is going to have the appropriate nucleotide to pair with the ones that we've already said. And so it'll start from the 3' end, and it'll be thymine first, and then guanine, and then cytosine, and then adenine. So 5' and 3' just refer to whether it ends with the phosphate group or with the ribose. It's kind of like proteins have a C terminus and an N terminus. Nucleic acids will have a 5' and a 3' end. And they're going to run opposite to each other. And so you see the adenine is the 5' end of one strand, and its complement, the thymine, is the 3' end of the other. And these DNA strands, not only do they connect all of their nucleotides, sometimes we'll call them base pairs because the nucleobases pair up together. So not only do they link together, but they'll also twist and curl around each other in a shape called a double helix. And if you look at this double helix, you can see that there's one groove right here which is more narrow, and there's a bigger groove right here which is wider. And so there are different proteins and different cofactors that will bind to the narrow groove or the wider groove. And every bit of the structure of DNA is really important to how it functions. So when DNA is all zipped up like this, all the base pairs stuck together, it's actually not going to be doing anything. That's like how it is stored when it's not in use. Oh, sorry, let's do a little bit of practice, right? So if I have a nucleotide sequence of 3' G, T, T, A, A, C, C, G, 5', what would be a strand that is its complement? It'd have to start with 5', then it would be C, A, A, T, T, G, G, C, 3'. Okay, so hopefully that's pretty easy to figure out. And then just to remind you, the RNA is going to replace thymines with uracil. So if we had a DNA sequence of the 3' GTT, AA, CC, G5', it could match with an RNA sequence, which was CAA, UUGGC 3'. So those Thymines would be replaced with uracils. Okay, so DNA is going to have to do two main functions. One of them is replication. Every time a cell divides, every new daughter cell needs to have a complete set of DNA. And so a part of the cell division process is to unzip the two strands of the DNA and replicate them both. Each strand is going to have a new complement manufactured piece by piece, and until you have two complete new sets of DNA. And a microbiology class can tell you about what happens from there as far as the steps of cell division and whatnot. But for us, we just need to know that there are specialized proteins that will grab onto DNA, that will begin to unzip them or to break those nucleobase hydrogen bonding interactions. And then one protein will do the unzipping, and then it will be followed by proteins that will replicate the DNA, that will construct a whole new strand of DNA to complement the now lone individual strands so that they each have their full complement. It's a pretty amazing process. So that the replication is still not getting at the meat of why DNA is important. That's just how we propagate DNA to new cells when the cells divide. So what's the point of DNA? And that is for translation ultimately. So transcription is when you take DNA and you will manufacture a strand of RNA which is complementary to it. And the reason that needs to happen is because DNA does not leave the nucleus, but all of the rest of the important organelles of the cell are going to need the information that DNA provides. And so RNA is like making a carbon copy of the DNA. The RNA is able to leave the nucleus and enter the other parts of the cell and do the work that needs to be done. So transcription is going from DNA to RNA. And it's just like DNA replication, except the proteins, instead of making a new DNA strand, they're going to make a new RNA strand. And after a portion of the DNA has been transcribed, it will zip right back together until it's needed again. So that's transcription, and then the next step is that the RNA will go to a part of the cell called the ribosome, and it will make proteins, and this is called translation. So translation is from RNA to protein. Transcription is DNA to RNA, translation is RNA to protein, and this is what's often called the central dogma of biochemistry. It's kind of, if you had to choose what is the one most important topic in biochemistry, it would probably be this process because it is so central to everything about how organisms live. The way that the DNA is able to provide information for manufacturing proteins is that every 3 nucleobases is going to be a code for a new amino acid. So the DNA gets transcribed to RNA, and RNA is going to have uracil in place of thymine. So you see here a lot of U's. And then that RNA is going to go to the ribosome and be read from beginning to end. And you can think of it kind of like Morse code or like any other code, or frankly like written English. Every 3 nucleobases is going to represent a new amino acid, and the ribosome is going to take that amino acid and bind it to the next one and the next one and the next one and the next one, and it churns out a protein out the other side. So you can see here a table. Again, there's no way you need to memorize this table, but hopefully you can sort of recognize how to use it. So on the left side we have the first position, U, C, A, and G. So that'll be the first of the 3 nucleobases. And then the second position is on the top. Those are the columns, U, C, A, G. And then finally within each of those cells, the 16 cells that represent the combinations of the first and second nucleobases, then you'll have the third position, U, C, A, or G. So for example, if I have first a cytosine and then an adenine and then a guanine, I'm going to take the second row, that's cytosine, the third column, that's adenine, so that's Ca, and then I'll look at the very bottom of that square for G, guanine. CAG codes for a glutamine amino acid, and you'll find a place for all 20 amino acids there. I don't know why or how important these redundancies are, but you'll notice that there are many different ways to code for serine or for proline or for glycine. Some of them have different combinations that are not adjacent to each other. So serine is in that first row, second column. That kind of makes sense that it's like the third nucleobase doesn't matter. But there's also some serine over in the adenine guanine square. So that's totally different in both the first and the second nucleobase. Anyway, I think it's kind of fun to look at this and think about it, but you could probably go mad trying to find patterns and things. Also, you'll notice there are 3 very special combinations. UAA, UAG, and UGA are all called stop codons. And if the ribosome is working and making a protein and churning out amino acids, once it reaches one of those 3 combinations, it will stop. It will disengage and release the protein, and it's done. So that's like the period at the end of a sentence. It signals to the cellular machinery that we are done. I want to say a quick word about RNA. We've been talking about it generally, but there are actually 3 different kinds of RNA. Mostly we've been talking about what's called mRNA. mRNA is the short strand of RNA that is transcribed from a section of DNA, carries that information out into the rest of the cell, and that is what our mRNA is. Well, there's also tRNA. So tRNA is going to be out in the cell, and it's going to, on one hand, grab onto an amino acid, and then on the other hand, it's going to have 3 nucleobases, and that is how these combinations of uracil, cytosine, adenine, and guanine are going to code for a particular amino acid. The mRNA is going to have, let's look at the upper left corner, right? The mRNA is going to have UUU. Well, somewhere out there is a piece of tRNA which has TTT, thymine, 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 and it is going to grab onto a phenylalanine. And it's going to come and bind to the mRNA and hold that phenylalanine just in the right position to form a peptide bond with whatever comes next and continue making the protein. So tRNA is just a short little 3 nucleobase sequence. It's called an anticodon because it is complementary to these 3-letter codons that are part of the mRNA. But then lastly, there's rRNA, or ribosomal RNA. And the ribosome is the machinery that makes the proteins, and it is also made of RNA with some other proteins. And it kind of raises a chicken and egg sort of a question. basically it looks like RNA is the type of molecule that was used to make all other types of molecules, and so it's kind of the progenitor or the basis of all the other biochemical processes that we have. So here's a little table summarizing it, right? DNA, it's double-stranded. It has A, T, G, and C nucleotides. The ribose has its oxygen removed from the second carbon. It stores genetic information. That's its whole purpose. It will be replicated upon cell division and transcribed in order to actually get that information where it needs to go in the cell. mRNA stands for messenger RNA, and it has uracil in place of the thymine, and it is going to be that short transcribed message of the DNA that then goes out to the rest of the cell. rRNA, or ribosomal RNA, forms the actual structure of the ribosome together with proteins, other proteins, and the ribosome is the cellular machinery which makes these proteins. It is RNA just like the others. It's single-stranded. It's got nucleotides, U, A, G, and C. And then tRNA, transfer RNA, are those little short anticodons, the 3 nucleotides that are going to sort of translate from RNA speak to protein speak. They grab onto a protein with one hand, and then they're complementary to the mRNA on the other hand, and they lock things into place. I guess I should have gone. I should have gone on the internet and found you a good video. Maybe I'll send you a link to one, because there's no point in me violating copyright and showing you a video in my video. But there are really great animations that show this process in a much better way than I can describe it with my own words. And I'll admit I found the at least the static pictures in the textbook to be a little bit wanting as far as explaining this process, which is extremely fascinating and complicated. So I'll just end my lecture here, and I'll send you an email tonight with a link to a good video animation that shows the process of transcription and translation. So, and that's it. I guess I'll go and write your final exam now or something.